Wise athletes everywhere don't just want better health as a foundation for better athletic performance. We want more life, a longer life filled with the physical activities we love, which requires more than mere wishful thinking to obtain. The hunt for more life is an old story, but now Ora Biomedical is boiling the ocean, searching among 1 million chemicals and combinations of chemicals lying in plain sight for clues to massive health and life extension. Today, in episode 117, Dr. Mitchell Lee of Ora Biomedical gives us non-scientists an update on the million-molecule moonshot project and explains how the WormBot AI platform can work to find solutions for human beings. And he also explains how you can support this effort as I have done by sponsoring a test of a single drug or drug combination. I sponsored curcumin, so wish me luck. In addition, Dr. Lee and I discussed my question of how to be bold but safe regarding the high wire act of using off-label pharmaceuticals and over-the-counter nutritional supplements to get a fast start on my and maybe your personal pursuit of extended healthspan. This question has been burning my brain as I personally search for a path that avoids the slippery slope of wishful thinking, marketing overpromises, and the dangers of combining too many powerful chemicals. All right, let's talk to Dr. Lee of Ora Biomedical. Dr. Mitchell Lee, welcome back to the Wise Athletes Podcast.
Great to be back.
Well, fantastic. Hey, I can only imagine how busy you are with your million molecule project. And now this new thing, this sponsor a compound program. Very exciting. So thanks for making some time for us.
Oh, that's great. Yeah, it's been a lot of fun. The field continues to move fast, and we continue to find interesting things.
I want to actually hear more about that. I have two goals for our chat today. One is to hear more about this million molecule challenge, how it works, why it matters for people like me interested in improving my health and extending my healthy time in life, how the program is going. I keep hearing that you guys are finding things. Ooh, what can you tell us? Okay. And then the second thing I'd like to get, which is not maybe exactly your wheelhouse because you're not a clinician, but you are a smart dude who's up to his eyeballs in the aging science. And so I kind of want to get into some of this question of how can people like me be bold without being stupid as we resort to adding chemicals into our lives, supplement to our diet and our other lifestyle interventions. What do you think? Can we talk about those things?
Those sound great. Yeah, absolutely. Right at the cutting edge of what we're doing and how it gets implemented to help people out.
Excellent. Just to make sure the audience understands your credentials here, can you tell us how did you come to know so much about the science of aging?
Oh, sure. Yeah, I got interested in the biology of aging when I was figuring out what I was going to do for my PhD doctoral work. I was always interested in questions that I didn't realize were questions.
Ah.
The question of why we age. It just seemed like a natural, weird, just random process. Didn't seem like it had any kind of regulation associated with it. But as scientists were finding out around that time, there is a lot of genetic regulation that establishes how the molecular and cellular changes that actually drive aging manifest. And you can change how quickly or slowly those molecular and cellular changes begin to happen that drive aging. And that leads to extending healthy lifespan. It also leads to fighting all manner of different chronic illnesses and diseases. It's actually surprising that targeting aging has such a broad impact on our health and in the context of fighting disease. So as I learned more and more about that, I was hooked. Did my research at the University of Washington, where I looked at connections between cancer biology and the biology of aging. And then I did some postdoctoral work where I asked how natural genetic variation impacts how well these lifespan extending drugs work. And now I am at Ora Biomedical. So we're a standalone company that's focused on developing the very best small molecules to extend our healthy lifespan.
Well, that's great. And you work with some names that are very well known in the space, Dr. Kaberlein and Kennedy. Is that right?
Yes, absolutely. Yeah, I did my research with Matt Kaberlein at the University of Washington. We've continued to work closely together. My postdoc work was with Daniel Promislow, who's really a leader in the evolutionary biology of aging and
Hmm.
systems biology of aging. Brian Kennedy, these other sort of, prominent pioneers in the field. I've been really lucky to kind of receive mentorship from them as well.
Well, that's great. And they are the two gentlemen that I mentioned. They're both at or somehow associated with Ora Biomedical. Is that right?
But yeah, Matt Caberline is our board chair. So he's one of the key leaders. He continues to help us out and promote Ora Biomedical. Brian Kennedy's on our founders team / science advisory board. So he's also a key person that we think about. Brian, he has done a lot of great work thinking about how to get natural products into the hands of people who it can benefit. So
Great.
he's already done some really interesting clinical studies to move things like alpha ketoglutarate, some of those other kind of natural products forward.
Well, good. So tell us about, let's shift into talking about Aura Biomedical and your million molecule project. We talked about that a lot in our last episode that we did together earlier this year. And so I wonder if you could just give us a brief for the new members of the audience, what is Aura Biomedical? And then tell us about your million molecule project. How is it going? And what can you tell us about the things that you found?
Excellent. Yeah. So the promise is small molecules that can target and change the rate of aging and extend our healthy lifespan. The problem is that for the last 50, 80 years of scientific research, we have not done a very good job of combing through chemical space to find those small molecules. We've found things that show the promise, but we've by no means found the best of the best. And, I kind of liken this, the analogy that I'm starting to favor is kind of mining chemical space.
Mm.
If you think about mining through the ground, looking for precious metals, you want to cover a lot of ground.
Mhm.
Right now, over the last 50 years, kind of scientists together have been collectively digging with their hands and hand spades to find these interventions. We've built a backhoe. We've built something that will comb through chemical space quicker than anybody else on the planet. We can get to and screen through compounds, find the interventions that extend lifespan, figure out how to combine those together to make breakthrough therapeutics. We can do that faster and quicker than anybody else right now. So the million molecule challenge is sort of the goal that has emerged from that. Right now, the largest publicly available database of longevity interventions has a little under 1,100 small molecules within it. That's not to say that all of those molecules extend lifespan. That's just the domain of space in terms of the tests that have been done. By screening a million, we're going to surpass that by orders of magnitude. We can do that in a few years,
Hmm.
where this has been the collective efforts of decades to get this work done.
Hmm. And now people like me can sponsor my favorite chemical. And what is the limit? Is it like FDA approved drugs, grass, generally regarded as safe? What's the set of things that people can choose from in order to sponsor a test? Tell us about that program.
Yeah, absolutely. So we've just launched this a few weeks ago. So what you can do, anybody on the planet can go to our website, orabiomedical.com, go to the store, and then they can pick from a list of compounds that we've made available, and you can sponsor longevity tests. So this is a way to, help finance the Million Molecule Challenge. This is also a way to build this world's largest small molecule intervention database. And what's really cool about this project in particular, the sponsored compounds are going into an open access database.
Hmm.
So we are going to make this available for the whole world to benefit from.
Hmm.
And the last part of this is, it's really kind of exciting, innovative citizen science. There's really no other project where anybody off the street can talk to a world leading scientist and say, hey, I want you to do this, test this thing out, test this combination. So we've been really, the more we talk about it and the more we kind of think about what we've created here, it's really kind of neat and revolutionary from a couple different ways. We're excited about it. But to your point, what is available to sponsor? Right now, we have it's the set of FDA approved compounds, and we're going to be slowly building in more molecules to that. So, natural products is going to be one of the next libraries that we make
Hmm.
available. So anybody can go in, select from their this list of compounds. You pay it's $100 to test one compound. It's $200 to test a pairwise combination. And right now we're getting these orders in. We're going to start up the experiments over the next month. And by around the end of January 2024, we're going to start releasing those results. So that's going to be not just a leaderboard. So, what you can do is you can say, I think this is going to be a top lifespan extending intervention. And we're going to put, rank order the best interventions that are identified from this project. So you can be the person who proposed the experiment that found this new groundbreaking intervention. We're also kind of celebrating the international scope of it as well. Mm-hmm. Right now, we have sponsorships from over 17 countries.
Wow.
And that's really from just grassroots, sponsorship and promoting, and with the small microphones and megaphones that we have available. We've already been able to get this global reach.
Fantastic. I have to say, I have not been able to affect the selection of a scientific project since I was in high school. So it's really quite exciting that I can participate in this. So thanks for doing that. I mean, what a wonderful idea, really. Whose idea was it? Because it is brilliant.
It was just sort of a natural thing that came together. So, we were thinking about the scope of what we could accomplish. That led, Matt, Ben, our CTO, and myself to kind of come upon this million molecule challenge.
Yeah.
We were promoting that for a little while, and then we were thinking how to just grow it. And pretty naturally, this came about to be like, well, what if we let people sponsor these things? Is there interest for that? There's really no precedent for it. So there was nothing we could go out to and look and see what kind of enthusiasm and interest we could generate. But, as we're out talking about what we do at Ora Biomedical, you always get people who want you to test, different things. My emails, I always get emails from people randomly. And so this is a way to sort of capitalize on that momentum, or really give that momentum, redirect it in something where people can get excited and see some downstream results from it.
I totally agree. That makes perfectly good sense. And in some cases, people are like complaining like, well, why aren't you doing this? And why aren't you doing that? It's like, hey, pony up, man. Put your money where your mouth is. You think that's a good one? Let's do it.
Yep.
All right.
Yeah, absolutely. We're excited. We are your scientific team at Ora Biomedical. Put us to work. We'll test what you want to test. You got the money. We got the time.
Okay, great. Well, good. So let's transition into, a topic that I think, it's kind of common sense that the question would come up, but I want to give you a chance to talk about it. And that's this: why should lay people think that the results of tests on worms is useful, helpful for learning about longevity and health span in people?
Yeah, two ways to think about that. One, just focusing on the biology. The things that break down with aging are highly conserved molecular and cellular features. So those molecular and cellular features are shared from worms all the way to humans. Things related to proteostasis, things related to mitochondrial function, how we perceive and respond to nutrients as they're coming into our organic system. These are things that, that's not a uniquely human problem. That's an animal problem. So this was, these kind of pathways and ways of perceiving this were established and sort of evolutionarily honed hundreds of millions of years ago. So really all animals have these basic molecular and cellular features that change with aging. So that's one answer.
Okay.
The other answer is more functional. If you look at the things that extend mammal lifespan, the best two are dietary restriction and treatment with rapamycin. Mm-hmm. Those are things that work in every organism we've tested it in, going beyond even animals into fungus, single-celled yeast. You can extend their lifespan from dietary restriction and rapamycin treatment. Wow. This is the same story that plays out from yeast, single-celled organisms, all the way to humans. So in terms of that conservation of intervention function, we see the same kind of benefit. So worms are actually a great system to comb through this chemical space.
Great. Okay. Related to this, when you're looking at the effect that the various chemicals has on worms, obviously you would be looking for the effect on lifespan. But it would seem to me that, in fact, I've heard that there are ways to improve lifespan that are negative from a quality of life perspective. And so, is there something of what you guys are doing that could be used in assessing the chemical interventions in terms of not just extending the life, but extending it in a way that would suggest that it would be good for humans too? Like are you assessing like movement quality and decision making, or any things where you could infer something more than just they stayed alive?
Yeah, absolutely. No, 100% the case. If there's an intervention that extends your numerical lifespan, but you're in a low quality state through that benefit, then it's not really a benefit. It's not really what we want.
Yeah.
At Ora, our system allows us to screen things for lifespan, but also numerous different health measures. We're taking
Okay.
videos of these animal populations throughout their entire lifespan. So
Hmm.
we can look at things related to movement, behavior, how they socially interact with each other,
Hmm.
and numerous different features. So our CTO, he is developing what we call health span clocks.
Hmm.
So these are things that you take a lot of different phenotypes, independent measures, you summarize those together, and they're actually predictive of lifespan. So we have about 16 different features right now related to health and movement and behavior that we track along the lifespan. And we can do some really neat stuff looking at, a given compound. You give them a treatment with it, and numerically they could be, about like 70% of the way through their lifespan, but they look like they're, teenagers or young
Hmm.
adults in terms of their movement and behavior. We can quantify that very accurately.
Hmm. Well, that's great. I guess I'm not surprised. You guys are very thorough. But yes, we totally want things that improve the length of our healthy time in life,
Yes.
not just to be, comatose for a lot of years. Well, and I know that you said that the public program where I could sponsor chemicals is limited, although you're expanding it. But I wonder, in your general million molecule thing, is there anything currently, or maybe an intention for somebody to try to duplicate the rapamycin story? Like, is there somebody going to go dig up in the dirt on some other island, like the way they found rapamycin on Easter Island?
Yeah, so that is a possibility to go out, explore the environment, and basically do, this kind of fundamental drug hunting. We don't really have to do that also, especially with things like mTOR inhibitors. So once mTOR inhibitors kind of found their way into the clinic and people started to see the efficacy around it, people
Hmm.
started to design a lot of different things that impact that same mechanism. So there
Hmm.
are dozens of these mTOR inhibitors that have been developed now. Nobody has really systematically went through and asked how they impact lifespan. That rapamycin improves healthy lifespan through mTOR inhibition is great, but I don't know, it's very unlikely that that is the best absolute way to target mTOR signaling. Or
Hmm.
are there other things that combine with mTOR signaling to get us even a better benefit?
Hmm.
So we've done a little bit of that work, and we've actually found it's a dual kinase inhibitor. So it impacts TOR signaling, but it also impacts some other signaling pathways as well.
Hmm.
And it works twice as well as rapamycin at extending lifespan in the animals that we test.
Wow.
Yeah, they're out there already, and there just hasn't been anybody to kind of turn over those stones yet.
Yeah. Okay, well, good. Glad to hear that. But it seems to me, I guess, related to this, that mTOR, short for mechanistic target of rapamycin, we didn't even know about it until we found rapamycin and we tried to understand, well, what was it doing? So, what other cellular mechanisms are out there that we don't know anything about? And we won't until we find something that has an effect that we didn't expect. And maybe you don't have to dig in the dirt to go find it. maybe it is stuff that's run off the shelf. It would seem like you'd have to look at things that you would have no reason to expect there to be a benefit and just see what happens.
Exactly. Yeah, yeah. No, too much of the biology of aging has kind of we've kind of canalized our thought a little bit. We've kind of put blinders on a little bit when we think about things like the hallmarks of aging. There are these kind of 12 fundamental functions that seem to change with aging. It's great to have that conceptual unity and to
Yeah.
kind of basically, summarize 50 years of research. That's excellent. But what it kind of inadvertently got people to think is that we've solved aging, or we've figured out that these, in fact, are the only 12, that there are no other features. And that's incorrect. Yeah, there are other mechanisms out there that we have not begun to understand that influence the aging process. And yeah, the only way to get there is to do this kind of large scale discovery approach where, yeah, just like what you said, instead of looking at the things that we think work, look at the things that you have no idea and just really start combing that unexplored, the dark matter of chemical space.
That's a good way of putting it. Good. So you mentioned the hallmarks, and I was thinking about that myself. It would sure be a shame if everything we're studying is somehow related to affecting mTOR. Because if I've already got a way of dealing with that, then I think the upside to finding a better way of dealing with that maybe is limited. So are the things that you're testing, are you expecting that they're going to be affecting multiple, more than just mTOR?
Yeah, we anticipate them going beyond mTOR, beyond even nutrient signaling as an even broader mechanism. So, things like insulin signaling, AMPK signaling, TOR signaling. If you're in the biology classroom, they treat those as separate, discrete pathways, but really in the cell, they're integrated into one network.
Hmm.
And we've seen that small molecules that change how that network functions are enriched for lifespan extending things. So that's sort of one general category
Hmm.
of lifespan extending intervention, are things that impact the nutrient signaling axis. There are things beyond that. There are things other than that. Or once you've optimized this nutrient signaling axis, what is the next mechanism to then target to get an even bigger benefit? If you think about kind of correcting each issue in sequence, can you get to even greater improvements in lifespan?
Right. So it's not just touching on different mechanisms or hallmarks, it's the sequence of how you address them is important as well. It's pretty complicated.
Yeah, yeah. Once you've solved one set of issues, what is the next set of issues you would have to deal with? And that's
Right.
one way to kind of think about that. And we don't understand the sequencing of biological aging like that really at all.
One dimension of complication comes from the fact that the people who are most interested in the results of this work are people who their bodies are already older. So finding a solution that if you used it once you were 21 years old for the rest of your life, it would dramatically slow your aging. Well, that's cool. My kids can benefit from that, but it's too late for me to do that. I can't unwind time. So that's what makes rapamycin so interesting, is that it seems to have a benefit even when you're already older. I think people hate to say that it makes you younger, but it improves function. It recovers more youthful function in some ways, which then in turn makes your immune system work a little better, which in turn maybe makes your rate of aging slow down. Anyway, the key is that it is helpful for a person like me, as opposed to maybe more of a calorie restriction type benefit where you really kind of have to start at the beginning to get the benefit. I was wondering if the topic has come up of this approach would seem to be exactly the sort of thing that the FDA would like to have for its GRASS list. There's this list of things which are generally regarded as safe. And pretty often we find that there's things on there that they're not that safe. And then we got to pull them back off after millions of people have been eating it for, tens of years, or longer. Like trans fats used to be on there. And then certain flavorings have been on there and have pulled off. And now there's a variety of things. Red dye number 3 is kind of in the news a lot lately, California banning that, but that's on the list. And so Has there been any discussion of maybe using this approach for testing things which are going to get into the food supply or are already in the food supply that people are worried about?
Yeah, no, that's actually something that we're going to be focusing on as we move forward. One of the main libraries we want to start looking at, we've got, the FDA approved compounds, natural products, these other grass compounds are
Yeah.
another big domain of that as well. So we're going to be putting that kind of library together so we can start doing some of that analysis. And yeah, it's kind of interesting to think about, these things that, are generally recognized as safe have people looked at longevity of organisms treated with those things or in being exposed to those things,
Yeah.
because it doesn't outrightly quickly result in pathology. Seems like it's been, these things have been then added to a list where they're recognized as safe, but we haven't done the longitudinal kind of studies looking at it. So yeah, we might actually refine what we think about as safe and healthy by doing these sort of fundamental tests of aging.
I'm glad to hear that. Related to that, because some of the issues that I think people worry about related to this grass list are things which are preservatives that are put into foods so that they're shelf stable and so processed foods. And the concern is that those chemicals have an effect on our gut microbiome, which more and more people believe is fundamental to our health. So the question would be, can we learn about human gut microbiome effects by studying worm gut microbiome effects?
Yeah, to an extent. So they do have a microbiome. The worms that we study, they actually eat bacteria as their primary food source.
Hmm.
So that ends up being, through time, their guts start to break down, and those bacteria start to kind of outgrow. So you actually get a sepsis kind
Uh,
of pathology in the worms
huh.
associated with that. Yeah, and there are really good studies that show some of the lifespan extending benefits of, I forget what small molecule it was, but it actually went through the microbiome. So the microbes exposed to the compound upregulated things related to folate synthesis, I want to say. And it
Oh,
was actually having enriched folate in the diet was improving the lifespan of the worms. So you can see these direct readouts on the microbiome with these kind of studies.
yeah, yeah. Acarbose is one that had, with mice had been shown to be life extending, and the thought was that part of that was an effect on the gut microbiome.
Yeah, that's actually a good friend of mine's research. He was
Uh,
pioneering that. Dr. Alessandro Bito. He's out of Matt Caberline's lab as well.
oh, nice.
Yeah, acarbose, it's something that makes it such that you don't break down the sugars, and it itself is sort of a long starch looking molecule. And that influences the microbiome composition. So,
Hmm.
the one thing that's artificial about the worm model is there's just one type of bacteria in their gut. In a mammal gut or a human gut, there's thousands, it's a very broad, diverse community of microbes. So when you start changing nutrients and starches and sugars that are in the gut, you're going to start skewing and changing the community composition based on who can most effectively eat and utilize whatever extra nutrient is in there. And so, yeah, Alessandro showed some really interesting work that acarbose wasn't just going through insulin signaling, which is what a lot of people were thinking, but it was actually altering that community composition at the microbiome level.
Yeah, well, that's fantastic. Yeah, thanks for the details there. Acarbose is a very popular supplement. I guess it's a pharmaceutical at this point, but still, it's a supplement that people are interested in for longevity purposes. But you said one bacteria in the worms. Is that because of the control that they're under in the testing, and that they only have one food, which is this single bacterium? But in the wild, they would have more diversity. Was that what you're saying?
That's exactly right. Yep. When they're in the lab, we give them the diet. We set the terms of the diet, and the diet is kind of the easiest microbe that we can culture in large quantities in a lab. So
Yeah.
they end up eating a particular version of E. coli, which, if you looked at the microbiomes of these animals in nature, very likely they wouldn't have much E. coli in there. That's a more of a mammal gut bacteria that
Oh,
you see. In the wild, their microbiome would be highly diverse.
okay. Well, that's interesting, and that makes me wonder about other things because obviously, you're doing a scientific study and you want as few variables changing as possible so that you can understand what the effect is of the one variable that you're trying to change. But in real life, people tend to be living in a real life and they have lots of variability between them because of their environment. And I wonder if there's any testing that you guys are doing or thinking of doing that would be beyond these chemical interventions? I mean, would you be like looking at the type of food and maybe the amount of food, the temperature, the light spectrum, all of these other things?
Yeah, absolutely. No, you've hit on something fundamental in terms of the challenge of translating things from the lab to humans. Even in a mouse study, they don't exercise a lot. They're not exposed to pathogens that might make them, give them a cold or make them sick. They don't have immune challenges. They don't really have to deal with changes in temperature and light. So it really makes it somewhat challenging to do that translation to something that experiences a messy, diverse world like humans out in nature. So that is a big challenge. How we can do those studies, you can start to more closely approximate those. You can create a more diverse diet for the animals to feed on. You can look across genetically diverse animals. So that's another thing. The animals that we have in the lab are all genetically identical to each other.
Mm-hmm.
And so you're missing a big chunk of how natural genetic variation is going to influence these compounds. So, you slowly add in complexity when you're doing these studies as you're getting closer to translating out.
Okay. Well, that's good. I guess the last thing that I'm interested in understanding about your project and how you guys are adding to the science, there's a huge sequence of events that have to happen to go from the kind of work that you guys are doing to where I can go to the store and buy something. And you're not doing all of it. But I think you are doing more than just the bot tests. Tell us, roughly speaking, what is the process of going from, a finding in your million molecule search to something further along the path of a person should take note and maybe do something about that?
Yeah, absolutely. So it depends on sort of the pathways that we go down. One is a disease specific pathway, one is an aging specific pathway. So as we're finding interventions that increase healthy lifespan, one of the first things that we do to translate that going forward is to test it in aged mice. So just like what you were saying is we want to test these interventions in a context where people who are already aged reap the benefits of it. We don't want to, kind of structure things around a continuous lifelong treatment of some small molecule. We really want it to be targeted and the dosing window narrow
Hmm.
and it have a good effect. So that's what our next step is testing in aged mice. From an aged mouse model, we would then be probably aiming into companion pets. Right
Oh.
now, the playbook for companion pets is being written by groups like the Dog Aging Project and Loyal and another company called Trivia.
Hmm.
So they're starting to figure out how to actually look at these lifespan extending compounds and measure aging and longevity itself as the endpoints instead of a more proximal disease state, which is kind of how you have to aim FDA trials right now, at least for humans. If we're doing that FDA route, we've got a panel of numerous different disease models in the worms. So find something that extends lifespan, test it across numerous different disease states, find those disease states that are best mitigated by that small molecule intervention, and then move into mammals from there.
Awesome. Okay. We're going to transition our conversation now into something a little more personal for me and people like me who are looking for things. But I think that what you just mentioned is sort of a part of that because at some point, you don't have to speak to it yet, but get ready. At some point of this process that you're describing, a human being is going to get wind of it and is going to say, I might take some of that myself for some benefit like that worm got, or like that mouse got, or that dog, or, wherever the chemical was along that continuum that you were describing. And so I'd be interested in getting your thoughts about that. Okay, so here it is. The second topic is this, and it's something we talked about briefly the last time we were together. And everybody knows that the older a person gets, the closer they get to the end. And so people like me, we start feeling pressure. We want to be proactive. We don't want to wait for the government to get around to telling us, yeah, go ahead and take this thing. This is safe and effective for longevity. So people like me, we are moving kind of ahead of the game. We're being bold, let's just say, and maybe we're being too bold, right? There's no guardrails for us, to make sure we don't do something crazy. So, we're like listening to people like you and other people, and we're talking amongst ourselves. And what are you doing? And, what's your blood work look like when you do that? And, things like that. So it's really a lot of uncharted ground. On top of which, there's this slippery slope of wishful thinking. The placebo effect is a powerful thing. And so, you spend your 30 cents and you took a pill and you felt better. Well, why did you feel better? I don't know. Well, and I don't actually mind. I think it's great that for 30 cents, I can feel better, even if it was all in my mind. I think that's great. What I'm worrying about, this is what I want to get at with your help here, is I worry that, I'm adding this one and that other thing. And I heard that this was good for worms and that was good for mice and this was good for dogs. And each one of them made a promise to me. And it's very hard to know whether those promises individually were true or not. There probably was some truth to it. But nobody can tell me anything about when I added all of those things together. Was that good? Do I get all those benefits added together? Do I get all those benefits multiplied together? Or are some of them offsetting each other now? Or maybe I'm hurting myself. This is the question that I'm interested in talking to you about. And I think maybe the way to start is to talk about that chart that's been floating around the internet for, I don't know, a couple of months now, of some work that I think you guys did, Ora Biomedical, with metformin combined with other chemicals that had sort of an unpredictable effect. Can you tell us about that?
Yeah, absolutely. So, yeah, that study that you're talking about, we tested metformin in combination with not just any old compounds, but other compounds from the FDA library.
Hmm.
Metformin is one of the most widely prescribed small molecules on the planet. So the likelihood that somebody will be taking metformin and something else from the FDA compound library is very high. So, what are the effects of two combinations of a drug? We tested this, and it's an Alzheimer's model. So it's kind of an interesting model. It goes through this early onset paralysis that's produced by upregulating one of the main things that goes wrong or accumulates with Alzheimer's. And we asked how these compounds interacted. Metformin on its own delays this paralysis by about 10 to 15%. It's highly reproducible. You can set your watch by that phenotype. What we then did was test metformin in combination with a set of different FDA approved compounds, just randomly selected from the library. A lot of cases we didn't see an interaction. Some cases we found an additive interaction. Like if metformin did 10%, the other one did 10%. This one, take them together, you get a 20% decrease, delay in paralysis. Other times you would get things like 100% delay in paralysis. It took them twice as long to become paralyzed under these two compound interactions. So a synergistic or multiplicative interaction there. But importantly and unpredictably, when you combine two things together also, you get a negative impact. You wash away all the positive benefits of metformin and you actually hasten paralysis. We saw that just almost as frequently as we saw an additive benefit, we saw that negative benefit.
Hmm.
So really unclear, and not something that if you looked at what the mechanisms these drugs were hitting, it wouldn't give you any insight. We couldn't have rationally predicted what would have given you a positive benefit or a negative benefit in that study.
It's kind of scary. I assume that all you big brains have gotten your heads together to think about, why is that the case?
Yeah, it's something that if you look at the literature, this is really the cutting edge of discovery right now, is doing these pairwise and three-way, four-way combinations of lifespan extending interventions to see what gives you those benefits and what kind of washes away those benefits. So we are still figuring that out. And it kind of speaks to what I mentioned earlier on about sort of the sequencing of aging.
Hmm.
Once you've already changed or altered something, what's the next thing that you want to do? What's the next thing that in that context might actually be deleterious? Or if you've got an intervention that's already tweaking one pathway and you add another thing that further tweaks that pathway, at what point do you start actually getting diminishing returns or even a negative response?
Hmm.
Because, TOR is actually a perfect example of this. You turn down TOR signaling a little bit, you get this benefit. You get a lifespan extending increase that's highly reproducible. You turn down TOR signaling too much, you'll kill an organism.
Hmm.
It is actually bad for it actively. So, hitting that Goldilocks range of not too much, not too little is something that you have to do to get the benefit. And we really don't have any good guidance to give people on that yet.
Wow. Okay. So on this point of mTOR and hitting it a little but not too much, something that I have recently read is that many of the most common supplements are hitting mTOR. And so it would seem like the more different supplements I'm taking, the more I'm hitting on mTOR, which maybe was good up to a point and then bad. And if I'm also taking rapamycin, which is a big hammer for mTOR, I think the odds of me hitting it too much just went way up if I'm taking a lot of different things.
Absolutely. And also, managing your diet well, being careful about exercising and exercising well. Once you're already, taking care of your diet and you're exercising, how you add in additional small molecules into that, nobody can give you advice on it. We haven't even done good studies in mice to look at exercised mice being given rapamycin or metformin or something like that, let alone do an exercise, dietary restriction, and then starting to layer on more and more small molecules. You're doing ultimately a large uncontrolled study on yourself. And the problem is you don't have a twin or an identical version of you that you're not doing those things to. So you have no idea whether you're getting a positive benefit or a negative interaction. The only thing that you'll be able to see is if something very bad, very quick happens. Otherwise, you're not going to get a signal out of it.
Yeah, right. This is my worry.
And like you said, that placebo effect. There is something to be said about psychology and stress and how we can basically create conditions internally that shorten our lifespan. Stress is a major thing that shortens lifespan. And
Mm.
feeling like you're doing something to promote your health and being proactive about it, the psychological position that affords, I could see that as being beneficial.
Mm.
But if you're already doing things like exercising, dieting, taking care of yourself, not drinking too much, you're kind of checking a lot of the boxes that are also the boxes that get checked with these small molecules in terms of the pathways that they hit.
Which again, getting back to the mTOR as an analogy of this type of impact is if I'm doing the lifestyle factors right, and on top of which I add exogenous chemicals, now maybe I'm doing too much.
Absolutely. Yeah. Yeah. And we don't know. And it could be, it could even be as nuanced as, somebody would benefit from it, somebody else would not benefit from it, and it has to do with very nuanced features of your genome and
Mm.
what kind of genetic background you have.
Mm.
It gets so complicated so quickly, and there is no cut and dry advice that you should do one thing or another thing or not that thing. We're just not even there yet. And what's so humbling is that we haven't even agreed on what the tests are to start saying whether we've done something right. Testing human blood work, looking at the things, what are the relevant features to measure to say that we are doing right by taking some small molecule. We're still writing that playbook right now too.
Yeah, it sure would be nice if we had some sort of biological age calculator that, we could plug in our blood work and our physiological function numbers and whatever, and it comes out and says, the thing you just added has lowered your biological rate of aging, and so good job on that.
Yep, absolutely. Yeah. One thing, just to throw in a plug for our sister company, OptiSpan, they work with us very closely, and that's
Mhm.
exactly what they're doing. So
Mhm.
they are writing the playbook for how to measure healthy aging changes in humans at the clinical level. And so right now they're building in, AI and getting some LLMs together and really doing everything they can to then develop a data set that can then become informative and prescriptive for people going forward who want to maximize their healthy lifespan.
Well, I look forward to hearing more about that. As you mentioned, that would really be the answer to figuring this out as to when can you do more and when are you doing too much. And so I wonder if, just because you're a smart person and in this area, is there anything that you could share, just your personal opinions for that matter, on things like the relative importance of supplemental chemicals versus lifestyle, the dosing, periodizing, taking things periodically but not continuously, and the number of different things that a person should feel worried about, exceeding, taking at the same time. What can you share?
Yeah, absolutely. I think I said it last time. I wish I knew a secret that I could tell you, but unfortunately, I don't. Diet, exercise, if you are being proactive about those and maintaining those, you're really doing, I think, probably 80 or 90% of the work right there.
Mm-hmm.
Continue to work with clinicians. If you do want to try to engage in these small molecules or natural products, do it under the closest medical supervision that you can manage, and just be really careful. Listen to your body. If it feels like you don't feel good or you're getting headaches or you notice that maybe you're starting to get colds more often or you feel sick more often, really take that seriously. Listen to your body if you're going to do these things. Work with clinicians, and even better, find the clinicians who have an idea and an understanding of geroscience,
Mm.
because not every clinician is dialed into this right now. If you went to the majority of clinicians and said, I want to take rapamycin, they would look at it and say, no, this is for kidney transplants. You don't want to take an immune suppressant. And you'd have to educate them on, what we've learned about rapamycin and mTOR. So you really got to find the clinicians who are thinking about geroscience in this way and work with them as closely as possible. That's the best advice I could give.
That's probably the right advice, even though I would much rather you told me the secret that
I
I.
know, yeah, it's a bummer. Yeah, I wish I could tell you something good. Yeah, yeah. But, on both ends of the spectrum, from discovery, development, all the way to implementation, we are figuring this out right now. And so we need support, we need enthusiasm, we need people to talk about it. And if we're all excited to move this science forward, that's going to be the best chance of getting it into people's hands in a safe, effective manner as quickly as possible.
Yep. Good. All right. Well, that's what I wanted to talk about, Dr. Lee. Thank you. I wonder if there's anything else you wanted to share or any more you wanted to tell us about how we could participate, be co-scientists in your million molecule search.
Yeah, absolutely. So yeah, go to orabiomedical.com. You'll see all over the website. There are different directions to go and sponsor the Million Molecule Challenge. You can get involved in this very direct way. In academic science, they call the people who tell you what to do in terms of experiments principal investigators. So you can be the principal investigator. You can put us to work as scientists. Let us do what we do best, and we can utilize your imagination to really do some exciting things together.
Well, fantastic. So if I sponsor, my favorite chemical, is it now off the list and it was just me sponsoring it?
That is how we have it right now. Yeah, it's
Okay.
one person can sponsor one molecule. We might end up building in retests once we have the initial version of the database put together, because that's more accurate with how science works.
Okay.
You don't just do something once. You got to show that it's reproducible and it happens
Sure.
numerous different times. So I'm actually really excited to see the clustering of data points once people recommend the same intervention multiple times.
Yeah. Well, my guess is that when you get the nutritional supplements added to the list, people will identify with things more closely, and people will be upset that their favorite is off the list, but maybe they'll find their second favorite then. Well, great. Well, Dr. Lee, thank you very much for taking some time on a Sunday. I really appreciate that. You have a great day.
Absolutely. Thanks, Joe.
Alrighty. Bye-bye. Thank you so much for listening into my discussion with Dr. Mitchell Lee, CEO of Ora Biomedical. You can find more information about Dr. Lee and Ora Biomedical in the show notes.